So welcome everybody to this session where we have with us our keynote speaker for today, Professor Michael Baudoin. I would start with this brief introduction and then he would start his talk. So Professor Michael Baudoin started his research career at Sorbonne University, where his PhD was dedicated to the linear and nonlinear propagation of acoustic waves in dense suspensions of microparticles. Then he joined the team of Professor Baroud at Ledec's École Polytechnique, where he studied the dynamics of liquid plugs in microfluidic networks. In 2008, he started to build his own team at Univ of Lille on subjects at the interface between microfluidics, acoustics, and microsystems. In particular, he studied how the presence of partially wetting particles can drastically modify the interfacial dynamics of fluids and how specific acoustic wave fields called acoustic vortices can be used to manipulate selectively microparticles and cells in a standard microscopy environment. In 2016, he became full professor and was elected junior fellow of the Institut Universitaire de France in 2019. Today, he'll be talking about extraordinary interfacial properties induced by partially wetting microparticles. Over to you, Michael.
Thanks a lot for the introduction. First of all, I would like to thank the organizer for the invitation. It's a great pleasure to participate in this event, even if I look forward to meet you all in person. So I will discuss some extraordinary interfacial properties induced by partially wetting microparticles. And in particular, I will discuss two different problems that I find particularly interesting. So I will start with reverse Safman-Taylor instability. So I guess that most of the audience know about Safman-Taylor instability. So Safman-Taylor instability that you can see on these movies is a very classical instability that you can get when you inject a less viscous fluid into a more viscous fluid, as you can see on this video. Here it's water that is poured into glycerol inside a HeLa cell, so a cell with a very small height. So while the refinement of this instability is quite complex, the physical origin is relatively simple. If you imagine a stable situation at the beginning where you have less viscous fluid that you pour into a more viscous fluid, and you have a small perturbation here, then you can see that the resistance to the flow in this direction due to the small perturbation will be smaller here than it is in the other directions. And hence, this small perturbation will be increased, which leads to an unstable situation. But of course, if you reverse the experiment and you take a fluid that is more viscous and you pour it into a less viscous fluid, for example, if you pour water in air, then this situation should be stable. And what we will look at is what happens when we add some microparticles to this problem. So the experiment is quite simple. It's the same. We have a HeLa cell, so two walls that are pretty close. But we will put some partially wettable particles on the surfaces. So we do it at small, the experiments are performed at small capillary numbers. So capillary number compare viscous effects to capillary forces, which means that capillary forces are dominant in our problem. The particles that we use are typically 15 micron size particles. They are some polymer particles. And these particles have a contact angle of 71 degrees, which means that they are partially wettable. So let's look what happens when we add some particles to this problem. Well, if we add a low amount of particles, so if we are at low particle concentration, we see what we discussed before. These situations is relatively stable. So we have some liquid here that is poured. Here you have air. and you see that particles are collected at the interface, but you have a situation that remain relatively stable. But if we have a higher concentration of particle, then we get this fingering instability. And we can have different situations depending whether we have very extremely low flow rate. So in this case, you see that the growth of the fingers is relatively random. Or if we increase a bit the flow rate, then we will have something with a more radial evolution of the fingers. So the question is, what is at the origin of this fingering instability in this case? Because if we look at the classical pattern, it should be a stable situation. So what we did was performing some experiments for many different parameters. So we changed the distance between the walls. We changed the concentration of particle, more or less concentration, and we changed the flow rate. And you can see that we can have a variety of patterns depending on these parameters with different size of the fingers. So the origin of this instability comes from the fact that we take some partially wettable particles. These partially wettable particles like to be at the interface between liquid and air to reduce the interfacial energy. And since we are working at low capillary number, this is really important. This is what will dominate the evolution. So at the beginning, as we have seen, when the cylinder grows, then the water cylinder grows, it will capture the particles. But if you have more particles on the surface than your interface can collect, then you get a problem because the particle would like to join the interface, but you have not enough space to incorporate them. So the solution is, of course, to increase the amount of surface that you have at a given volume of water. And to do that, of course, the simple solution is to create some fingers. To verify this hypothesis, what we did was pretty simple. We looked at the moment when the instability starts, and we computed a critical radius when the water interface, it will be covered by the particles. And this critical radius, of course, depends on the concentration of particles on both plates, but also on the contact angle of the liquid with the walls. And we compare it to experiments. Here is the critical radius when the instability starts as a function of the concentration of the particles that we measured on our experiments. And this is the experimental points for two different capillary numbers. And these are the model for two contact angle of the two extreme contact angle of the liquid with the walls, because we don't know it perfectly, because the particle, there is some pollution on the surface and it can vary between 0 and pi/2. And we see that indeed the instability starts when the interface is completely covered with particles. Now, the second question, we wanted to go further, and we were wondering what fixes the finger width. So to simplify the problem and to have a simpler geometry, we used this time not a cylindrical cell, but a square channel. And we performed relatively similar experiment, but this time we pour the liquid into a rectangular channel. So what we have is some square tube of 1 millimeter per 1 millimeter. And again, we are at low capillary number. And here is what happens. So we pour the liquid, as you can see here. So the liquid is here. There is some particles on the walls, and we will start the experiments and pour the liquid into the channel. And what you see is at the beginning, again, the interface collects the particles. And so it goes through here, it's just some glue to make the link between the tube and the rectangular channel. And you see that at the beginning, again, it's straight, but at some point, there is a fingering instability that develops. The liquid will be just on one side, and you will have an interface that is covered with particles. And you can do the experiments for a long time, and you see that the width of the finger is pretty stable as long as you have a given concentration of particles on the walls. So again, we looked at what fixes this finger width, and we established a simple criterion. The width of the finger will be fixed by the following criterion. All the particles that are encountered will be incorporated in the lateral interface. And with this simple criterion, we are able to reproduce the width of the fingers that we get depending on the concentration of the particles. And we have a pretty good agreement, again, between this very simple model and the experiments. So we show this simple experiment shows that a stable situation can be turned into an unstable situation by the simple presence of microparticles on the walls. And if you want to know more about this work, then you can look at our papers that we published in 2016 in Physical Review Letters. So now I will move to a completely different problem. The problem is everlasting bubbles that will resist drainage, evaporation, and nuclei-induced bursting. So the problem that we wanted to address is, can we produce bubbles which never pop? So it's the dreams of children. When you produce some soap bubbles, you all know that they will eventually break. And we were wondering, can we create some soap bubbles that never break? So when you have a soap bubble that is looked at with a color camera, and you see that after some time, you see some interference fringe here, which indicates that the film is thicker here than here. And this is simply due to the effect of gravity that induced drainage. And this is the main reason for the rupture of bubble, because the upper film will become very thin and very sensitive, either to evaporation, and we will discuss this later, or to nuclei-induced bursting, which means that any dust in the air can produce the breaking of the bubble. So what are the characteristic living time of a bubble? Well, it depends. Without surfactant, you will have gravity-induced drainage. And since without surfactant, you have a sleeping condition between the liquid and the air here, then this characteristic time will not depend on the thickness of the film, but it will just depend on the viscosity, the gravity that is at the source of the flow, and the characteristic length will be the radius of the bubble, because this is what fixes the gradient of pressure inside your liquid film. So the lifetime does not depend on the film thickness. And if you have a big bubble, you will have very short lifetime. So the only way to get such bubble with a long living time is to have very, very, very viscous liquids. When you add surfactants, the situation is more complex. First, the presence of surfactant will turn the slip boundary condition into a no-slip boundary condition. And this will considerably slow down drainage, because this characteristic length will be turned into the thickness of the film. And so the characteristic time will be increased a lot. But eventually, you still have some drainage, and the film will become very thin. But surfactant can stabilize very thin films. So it was demonstrated by Grosser that you can keep soap bubble for a very long time, as long as you suppress evaporation. So you are in a saturated atmosphere and you have no nuclei. So this old work in 1969, they succeeded to keep some bubbles for a very long time, but in a saturated controlled atmosphere and with no particles inside it. And what we wanted to do is to have very long lasting bubbles, but without all these constraints in a standard atmosphere, without any control of the atmosphere. So the question we asked is, how can we suppress drainage, evaporation, and nuclei-induced bursting? So for the drainage, what we did was to use our magical particles and create bubbles where surfactants are replaced by partially wettable particles. So here is the experiment. So we have some liquid. We put some microparticles on the top. And we create a liquid raft. And after some time, then we just inject a bubble behind this raft. So after, to cover the whole surface of the particle, of the bubble with particle, what we do is to just move it on the raft. And finally, we can take our particle and put it on a substrate. And you have here, it's really a bubble, but covered with some microparticles. So now let's look at the lifetime of this object. So this is the same object as you have seen before. The only thing that we did is to add some blue dye to see the evaporation of the liquid. And the experiment that you will see will last for 64 minutes. So it's accelerated a lot. And one important point is without the particle, the living time of this water bubble, it's just water with microparticles, will be very, very short, around 20 microseconds. So here is what we obtain. You see that the bubble keeps its shape for a very long time. And here you have seen it's popping. And you see that after it pops, you see that the liquid is, all the liquid is almost evaporated. To be sure that it was really evaporation that changed the color of the bubble, what we did is looking at the bubble mass as a function of time. Here is the point when the bubble pops, and you see that popping occurs almost when the bubble mass reaches a plateau here. And this plateau corresponds to the moment when all the liquid has evaporated. So we have suppressed drainage, and now what is responsible for the death of our bubble is really the evaporation of the liquid. So to verify this on more experiments, we did experiments on many different bubbles with different size and different ambient humidity. And of course, the more humid is the atmosphere, the longer will be the evaporation process. And so the longer will be your living time. And this is what we indeed observe experimentally. So another proof that the bubble is dry is that sometimes after the drying process, the bubble breaks. And you see that it really breaks like a sand pile, meaning that there is no liquid anymore in this bubble. So why did we succeed to suppress drainage? Well, it's quite simple. At the beginning, if you imagine the same bubble without the particles, then you will have a bond number that compares gravity effect to surface tension that will be large. But when you add some particles, then instead of having a big liquid film, you will have some small capillary bridge. And at the size of the capillary bridge, the bond number becomes small, which means that the surface tension will dominate over gravity and will enable to sustain the liquid and avoid the drainage of the liquid. But we still add the problem of evaporation. If you want to have even longer living time of your bubbles, you need to suppress evaporations. So how can you suppress evaporation? Well, by using a hygroscopic liquid. What is a hygroscopic liquid? It is a liquid that tends to absorb the water that is contained in the surrounding atmosphere. And why he does that? Because an example is glycerol, and glycerol has a lot of hydroxyl groups. And these hydroxyl groups, it tends to create some hydrogen bonds with water, and so to attract water at the surface of the molecule. And this is why they tend to absorb water. So we did many experiments with different glycerol water mixtures. So we just replaced water by water glycerol mixture with different water glycerol mass ratio. So from 0 to 0.9. And what we observe is two completely different behavior. One behavior here that you see is that is very similar. So here it's of course pure water. Here this is pure water, and here it's water with a small amount of glycerol. And what you see is that you have a behavior that is pretty similar to what we have with pure water. The bubble mass decreases, and it leads to the rupture of the bubble. But above a certain mass content of glycerol, you see that the bubble mass, if you have a small amount of glycerol, the water evaporates, but at the end it reaches a plateau where the bubble mass doesn't evolve anymore and doesn't rupture. And if you add more glycerol, then at some point you will have a stable behavior from the start. And if you even have more glycerol, it will be the opposite. At the beginning, the bubble mass will increase by capturing some water that is in the surrounding water atmosphere, and tends again to an equilibrium. And in fact, if you look at the glycerol mass ratio as a function of time, you see that all the curves converge toward an equilibrium mass ratio that depends on the humidity, ambient humidity. And with this the type of bubble, we were able to keep bubbles for more than 1 year, because our oldest bubble died after more than 460 days. And the reason has nothing to do with physics, it has to do with biology, because we had some glycerol in water and we didn't take any precaution to create these bubbles. And we saw that the bubble became green because probably some fungi developed inside our bubble. So how can we be sure that they were still liquid after this time? Well, what we did was puncture these bubbles, these long bubbles, and you see that this has nothing to do with the previous behavior. You have a liquid-like behavior because you still have some liquid between the particles. So can we predict the next thing that we ask is, can we predict first the mass evolution of this bubble, and can we predict whether the bubble will be stable or not, depending on the ambient atmosphere humidity and the amount of glycerol that we put at the beginning. So to look at the mass evolution, it's quite a simple process. The evolution of the mass will be driven by the absorption of water due to the presence of glycerol and the evaporation. So of course, evaporation will depend on the amount of water in the film of the bubble, which will be 1 minus the amount of glycerol. And it will be proportional to 1 minus the humidity, because of course, the more humidity you have in air, the less evaporation you will get. And if the humidity is equal to 1, then you will have no evaporation anymore. And you have, of course, a constant here that needs to be determined. For the absorption, it's the same. It's the opposite, sorry. The absorption will be, of course, dependent on how much glycerol you have inside your thin film. And this time, the absorption will depend on how much liquid you have in your surrounding atmosphere. And again, you have a constant that needs to be determined. But the beautiful thing is that we can determine this constant without any adjustable parameter, because the evaporation coefficient can be determined from experiments that have been performed on pure water gas marble. So in this case, of course, the absorption is equal to 0. You don't have any glycerol, and the evaporation will be Ke1 minus H. And what we did is finding the coefficient that gives the best results for all our experiments. And with one coefficient, we are able to get pretty good agreement between the solid lines here that represent the theory and the experiments that are the dots. But we have a second parameter to determine the absorption coefficient. And for that, we can use some experiments that exist that tells us that for at equilibrium, so if you wait a sufficiently long time, then there is a ratio. If you put some glycerol, you will eventually reach a ratio of glycerol on water that depends on the ambient humidity. And there are many experiments in the literature. So you have lots of experiments. And we can again take our simple model. In this case, at equilibrium, the evolution of mass is equal to 0. So absorption is equal to evaporation. So we will have that equal to that, and which gives us equilibrium glycerol mass ratio that will depend on, of course, the humidity and our coefficient Ka over Ke. So by fitting the experimental data, we can determine Ka over Ke, and we obtain this coefficient. And now we just have to take our model and compare it to all the experiments that we perform. And you see that with our simple model here, we are able to fit all the experimental data with quite a good agreement. And not only we are able to predict the mass evolution of our bubble as a function of time, but we are able to predict whether the bubble will last forever or die, depending on the initial glycerol mass ratio and the relative humidity. So here you have a diagram with the experiments. Here, in this case, you have systematic rupture. Here you have everlasting bubbles. And here you have bubbles in between, which means that we did experiment on a lot of bubbles. And in this region, of course, you have some bubbles that die and some that remain stable. And the line here is what we get with our theory. Of course, you have some error bar that correspond to these two lines, but which are quite in good agreement, again, with experiments. But to conclude, this is a new material, this composite material, and we did the experiments for a bubble, but you can use it for any type of experiments with interfaces. So here it's just a metallic frame, and you can create a soap film, and if you do it with a soap film, after 39 seconds, the soap film will break, as you see here. If you add just particles, no glycerol, then it will break again, but after 8 minutes, because again, what will be responsible for the breaking is evaporation, and you can see indeed that you have a quite solid behavior when it breaks. But if you add glycerol, then you can have film stable for months if your student doesn't kill the experiment in the meanwhile. So this one couldn't last more than 180 days just because there were some mistakes on someone pushed the film. But these experiments, we can even create some more complex shape like a pyramid here. And this one is still stable in our lab after 1 year. So with that, I just want to underline that the presence of partially wetting particles can completely modify the liquid-air interface behavior. I didn't have time to present all the results that we got, and we have also some nice results on some Armo bubbles in a liquid. But I think it's a very nice path to follow and study. And if you have some question, I will be very happy to answer them. Thank you for your attention.
Thank you, Mikhail. It was a very interesting talk. We already have a question, and more will be coming, I'm sure. This is by Arvind. He's asking, he first congratulates on your talk. Interesting talk is what he says. And he has two questions. First is, what does partially wetting mean for microparticles? he wants you to elaborate on that.
Yes. So just for this one, so what we mean by partially wetting particles is you can have particles that tends to be completely wetted by the liquid. So if you have, there is a parameter that is called the parameter else that tells you that if you can compute the interfacial energy of your particle and look whether it prefers to remain in gas or to be completely covered by liquid. So if it's if these are completely wetting particles, they will go inside the liquid. So you cannot keep them at the interface. So when we say partially wetting particles, it's just particles that will not be completely wettable and have some contact angles that are between, let's say, 0 and 180 degrees. Basically, the particles that we use are in between. They have a contact angle of 70 degrees. In these experiments, we didn't perform experiments on many different types of particles, but for another program we did. So of course, you need particles that like to be at the interface between liquid and air. So at some point you can lose this interesting behavior if they are wet too well. So if the contact angle is too close to 0, and you can have also different behavior if they are hydrophobic or super hydrophobic.
Second question that he has is, can the inverse Safman-Taylor instability also be observed when glycerol is moving in water suspended with microparticles? So the ambient fluid, I think he wants to say, is water with particles, microparticles.
In fact, there have been, after our papers, there have been papers that have been published where they try to redo an experiment similar with 2 liquids. So what they did was injected some particles, and this time they had 2 liquids. But I don't remember exactly the liquids they use. So I guess you can also get, yes, this kind of instability if you use 2 liquids and this microparticle. Yes, I would say yes. And I think there is a paper on that that was published, I think, 1 year after, something like that. Yeah.
I see. I would like to add to that question. I mean, I have the follow-up question that I have on that is, suppose if you have a viscoelasticity, some viscoelastic material or some non-Newtonian behavior, let's say, how would that modify Safman-Taylor instability criteria? That viscosity ratio would still be the criteria, or would...
what you must understand is that in our case, we work at very low capillary numbers. So it's almost quasi-static evolution of the interface. So what drives everything in our process is really interfacial phenomena and not the viscous and the flow process. It's not completely true because if I go back, as I told you at the beginning, when we change the flow rate, here I show you a first behavior where when you look at the evolution of the fingers, it's completely random because here it's completely quasi-static, which means that only interface matters. So then there is no reason that the flow will be radial or anything. So you can see that it goes in any direction randomly. If you increase a bit the flow rate, you are a bit in between. This time you see that you still have a radial evolution, which means that the flow plays a role. Okay. But it's still what matters the most is really the interfacial phenomena. This is what drives the fingering stability.
Mm-hmm. Okay. Arvind has another question where he's asking you, what is the stability against nucleation induced bubble bursting with microparticles? So
this is a good question. The thing is, we don't observe it because we keep our bubble for 1 year. So my guess is, since you have microparticle at the top, it's like an armor at the top of the bubble. And even if you have dust coming, first, there are 2 things. First, the dust might not come into contact with the liquid because they will come into contact with the particle armor. And second is that you have very strong capillary bridges instead of a very thin and fragile liquid film. So even if the particle came into contact with the liquid, I guess it will not break the bubble. So I think this is what makes the bubble stable. Of course, if you are able, this is what we did. We pinch the bubble. So if you go really through the air more, then you can break the bubble. But it does not happen with the dust that you have in the air, while it happens, of course, a lot when you have normal bubbles.
Okay. Mikhail, when you cover the particle or when you prepare the solution do you think it's going to be a monolayer of particles or a double layer because you have two interfaces when you
Yes, this is a good question. In fact, we try to look at it. What we see is that I don't think it's a monolayer. The reason is that it's not very spherical and nice particles. So it's particles with complex shape. So I wouldn't say that we have a monolayer of particles. But I don't think that you have, if it is your question, a layer of particle on both sides. But you can do it. What we did, it's on the experiments with the flat film, the last experiment that I showed. If I come back here, this is experiments that we did with this one. Yes, this one we did it on flat films, and if here we just create a raft. We take it from below and put it up, and we have particle just on the upper surface. But we try to put particle on both sides, and in fact, you can do it, and it works. But this is not what occurs when you do it with our process. So I will say you don't have, naturally on our bubbles, you don't have particle on both sides, but you have not a monolayer of particle on the upper side.
Mm-hmm. The reason why I'm asking is you can create a bubble which is thicker than what normally the thickness that you get in case when you don't have any particles.
So, yes. Our dream was to make them fly, to make this funny, but in fact they are too heavy, so we didn't succeed to make our bubbles fly just because indeed they are much thicker and
Yeah. So did you look at the particle packing, how it is like? Is it following a hexagonal pattern or is it just purely random or something?
Again, I think this will be a really interesting study to do if we had some really normalized particles. We wanted to do this, but because it's better from the physics point of view to have a more controlled system. But the thing is that we use a lot of particles, as you have seen, to make the experiments, and calibrated sphere exist. They are first very expensive, and generally they are not hydrophobic, partially wettable, which means that you need to treat them. So we started to do that, but it's quite a long process for microparticles to get them with the good hydro properties. So we would like to look at that, how they are arranged. And there is a mystery that we didn't answer and are working on, is normally you have a pressure inside the bubble due to Laplace overpressure. And the question is how this overpressure disappears, does it disappear, and how it disappears if it does, and how the bubble can keep its integrity if it disappears. And so we are trying to answer this question. This is a really tricky question to understand, and for that I would like to use some very controlled particles, because I would like to see. There have been experiments performed. There were the physical review letters with bubble inside water that remain stable for a very long time. And they were wondering why they do not vanish because of not evaporation, but the dilution of the inner gas inside the water outside, like it does for every bubble. And the reason was that the particles were reorganizing in a way where there were no curvature anymore of the liquid between the particles. So you had no Laplace pressure anymore. And this is something that we would like to look at with very controlled particles. So we didn't do that, but this is indeed something very interesting to look at.
Yeah. We have one more question from another person from the audience, Ram Krishna. He's asking, were particles spread uniformly in Halley-Shaw cell?
Yes, yes. It was. So what we did is try to spread them as uniformly as we could. So we spent a lot of time on that, and basically we were not able to predict what was the concentration. So we measured afterward. So I won't say the concentration was completely uniform, but it was. If you look, especially on the experiments in the rectangular channel, we did some pretty good job, I guess, because you can see here that you have on this movie that you have a pretty uniform repartition. So it's not exact, and you see that you have some fluctuation of the fingers that are probably due to the fact that you had more concentration or less concentration of particle at some point. But it's difficult, I think, to do something very, very homogeneous and precise. Yeah.
Right. Yeah. Yeah. So, we have no more questions currently in the Q&A section, but I'm sure there will be more coming. So, if you're Mikkel, if you're attending the talk, then maybe you would get to see them in the Q&A section. If you can respond, or I'll email it. I think best would be that I can email them to you.
Perfect.
Right, and you may respond to them. It was a fantastic talk. It was really nice to hear about these two problems that we discussed. And on behalf of the organizers, I would like to thank you for this very nice presentation.
Thanks a lot for the invitation.
Thanks. So with this, we would close the session. And thank you, Mikkel, again. Yeah.
Thank you.
Thank you.
Bye-bye.
Okay, bye.